Hello, welcome again to Summit Biochemistry Hall, your go-to place for mastering biochemistry, one concept at a time. I'm your host, Dr. Juliet. Do you know that the body does not rely entirely on carbohydrates for its supply of glucose? The liver and a certain part of the kidney, which we call the renal cortex, can actually synthesize glucose from non-carbohydrate sources like lactate, glycerol, and even from glucogenic amino acids like alanine, glutamate, asparagine. Today, we shall see how the liver is able to synthesize glucose from non-carbohydrate sources from scratch, which is basically to help maintain a constant blood glucose level during starvation or fast. Now, gluconeogenesis is basically almost a reversal of the glycolytic pathway, except as step 1, 3, and 10 of glycolysis. Now, looking at the steps, which are 11 here, we see that the reversible steps of gluconeogenesis is exactly the same as the reversible steps in glycolysis. Step 10 is bypassed by these two steps. Step 10 of glycolysis is bypassed by these two steps. Step 3 of glycolysis is step 9, and then step 1 of glycolysis is step 11 of gluconogenic pathway. How does the liver make glucose from scratch? The liver can make glucose from scratch, which is from non-carbohydrate sources, like lactate. Lactate is usually the byproduct of glycolysis in muscles and red blood cells because of insufficient oxygen supply in muscle during strenuous activity, and because the red blood cells have no mitochondria and so can only do anaerobic glycolysis. So much of this accumulates in the muscle and red blood cells and are transferred to the liver to be used for production of glucose. The liver also can make use of glycerol from the breakdown of fat reserves in the adipose tissue. The glycerol from the breakdown of triglyceride is acted upon by two enzymes, glycerol kinase to give us glycerol phosphate and glycerol phosphate dehydrogenase to give us dihydroxy acetone phosphate, which is one of the substrates of glycolytic cycle. And then this dihydroxy acetone phosphate combines with glyceride 3-phosphate in the course of gluconeogenesis to give us glucose. And then lastly, gluconeogenesis is possible also because the liver can make use of glucogenic amino acids like alanine, which is very much in the muscle. Alanine is deaminated by aminotransferase enzymes to give us pyruvate, which enters the glucogenic pathway. Lactate enters also after being reduced to get pyruvate, and glycerol enters at this point. Other enzymes, other amino acids like glutamine, glutamate, arginine, proline, histidine are all reacted upon to give alpha ketoglutarate, which enters the TCA cycle until it gets to oxaloacetate and joins this pathway to give us glucose. Now let's look at the basic steps of gluconeogenesis. Pyruvate, having been synthesized or obtained from lactate or alanine, is acted upon by the enzyme pyruvate carboxylase. This is a mitochondrial enzyme. So pyruvate at this point has to enter the mitochondria of liver cells to be acted upon by this enzyme with the help of ATP, adding a carboxyl group to pyruvate to give us oxaloacetate. And the oxaloacetate now is also acted upon by another enzyme right there in the mitochondria, phosphoenol pyruvate carboxyl kinase, to give us phosphoenol pyruvate. Now this is the gist. If pyruvate is obtained from lactate, there will be no need for oxaloacetate to be converted in the mitochondria. If lactate is the supplier of this pyruvate, then this reaction will go this way. Pyruvate is converted to oxaloacetate to phosphoenol before this comes out of the mitochondria. But if the pyruvate is supplied by any other source like alanine, then this phosphoenolpyruvate will be formed outside the mitochondria. Why? If lactate is produced, is the source of this pyruvate, NADH is already available here for this step of the reaction. So there's no need for NADH to be released from the mitochondria. So this reaction will just go this way. Pyruvate enters the mitochondria, produces oxaloacetate. Oxaloacetate is converted to phosphoenolpyruvate, and this is released in the mitochondria. But when the alanine is supplying the pyruvate, then oxaloacetate is converted to malate in the mitochondria. Malate comes out of the cytosol and is converted back to oxaloacetate and then to phosphoenolpyruvate, just to ensure that this NADH comes out of the mitochondria into the cytosol for continuation of gluconeogenesis at this point. Now, having made that clear from phosphoenolpyruvate, so that means depending on the source of pyruvate, this enzyme at this point can be a cytosolic isozyme or a mitochondrial isozyme. And then we see there that we don't need ATP. The energy molecule used here is GTP to give us GDP plus carboxyl. So you can see that the carboxyl group that was added here has also been removed here to give us phosphoenolpyruvate. Now that goes on to give us water is added to phosphoenolpyruvate. This step, this are simply reversal of the glycolytic pathway. So you see water entering again, still catalyzed by enolase to give us 2-phosphoglycerate. Now this is acted upon by phosphoglycerate mutase to give us 3-phosphoglycerate. And then from there we move to the phosphorylation of this to give us 1,3-bis phosphoglycerate by the same enzyme of glycolysis, phosphoglycerate kinase. And then from there we get our glyceride 3-phosphate from the oxidation, from the reduction of 1,3-bis phosphoglycerate to give us glyceride 3-phosphate. Now this is added to dihydroacetone phosphate. There is an adjoining by aldolase at point 8, step 8, to give us fructose 1,6-bis phosphate, also a glycolytic intermediate. And fructose 1,6-bis phosphate, which is up here, is dephosphorylated by the addition of water to give us fructose 6-phosphate. Now the enzyme here at this point is not a glycolytic enzyme. All of these we've seen at this point again, where there's a bypass of the glycolytic reaction, we have fructose 1,6-bis phosphatase acting upon this to remove a phosphate group from this to give us fructose 6-phosphate. And then this also undergoes isomerization, as we saw in glycolysis, to give glucose 6-phosphate by hexose phosphate isomerase or glucophosphate isomerase. And then lastly, again at this last step, glucose 6-phosphate is acted upon by water to remove its phosphate group to give us glucose, our end product. Now this glucose by the liver can then be released into the blood vessels to supply blood to other organs like the red blood cell, like the renal medulla, that cannot use any other source of fuel like fats. They cannot use fat, they cannot use amino acids, they just depend on glucose. And so the liver, through this pathway, is able to maintain a stable blood glucose level even during starvation or long-term fast. Now we've been able to show that gluconeogenesis is almost a reversal of the glycolytic pathway. It's step at this point, except at this step, at this step, at this step, and at this step. Now the step, this is step 10. The whole of this is step 10 in the glycolytic pathway, but now we see that 2 enzymes are able to change pyruvate to phosphoenolpyruvate during glycolysis, which can either be mitochondrial enzyme, or one can be mitochondrial and one certainly, but basically almost all the time, pyruvate carboxylase, this enzyme is a mitochondrial enzyme. Then we see the liver shuttling or the cell shuttling NADH for its reaction, either if it's coming from here or from here. I believe that we have done justice to gluconeogenesis. which we see is almost a reversal of glycolysis, and it's basically for the maintenance of a stable blood glucose level, even during starvation or long-term fast. Gluconeogenesis happens, begins immediately about 4 to 6 hours after your fast, and then peaks at about 24 hours later when the liver glycogen reserve must have been exhausted. It's been a wonderful time with you today, learning the gluconeogenic pathway in the liver and to a lesser extent in the renal cortex for the maintenance of a stable blood glucose level. Until I see you again, it's still Dr. Juliet, and bye-bye for now.